Hi, and welcome to my OCR A A-Level Biology revision session with me, Christine. So today's lesson I want to look at the mitochondrion structure and the link reaction. So let's just remind ourselves about the mitochondrion. So the mitochondrion is found within the cytoplasm, and the mitochondrion is a membrane-bound organelle because it has an outer membrane. That means that it has been separated from the cytoplasm, and that's important. Now, the next thing we need to know is it has an inner membrane. So the space between the outer membrane and the inner membrane is known as the intermembrane space. And the inner membrane has lots of folds in it, and that is known as the cristae. Now, that's important. The fact that it has lots of folds in this cristae is important in the fact that these folds provide a larger surface area, and that surface area is necessary for housing the proteins that are needed for the oxidative phosphorylation part in respiration that occurs within the cristae. Now, what we also note is the fact that these cristae are an extension means that this intermembrane space is very close together, and therefore within the cristae what you can get is what's known as a proton gradient building up. I will look at that in more detail in my lesson on oxidative phosphorylation. But it's important to note that the cristae is the point where oxidative phosphorylation occurs, and that is the place where you will find your electron transport chain. Now, the next thing we need to know is the mitochondrial matrix. So the mitochondrial matrix is a fluid-filled area. Now, that's important because if we look at this and look inside the matrix, we would find that there is circular DNA. Well, that should remind us that the prokaryotic distinguishing feature is that their DNA is circular. Now, we call the DNA inside the mitochondrial matrix mitochondrial DNA. So we use the small m and then DNA. Now, that is important when you're looking at module 6 and we are covering the genetic material and how your genome is inherited. Also, within that mitochondrial matrix, you find 70S ribosomes, another distinguishing feature which shows us that this once was a prokaryotic organism that was engulfed, because of that outer membrane, by a larger prokaryotic organism, which resulted in this membrane-bound organelle. And we call that the endosymbiotic theory. Now, one of the things they love to do in your exams, they love to throw an electron micrograph at you. So the key thing to remember is the fact that we can see the structure of the mitochondrion. If we can see the ultrastructure, we have to be looking at a transmission electron microscope micrograph. Now, the next thing you need to note as well is that you can clearly see the inner folds of the cristae within the diagram. So if they were to give you a different section, so what they're saying is, remember, this thin section has been sliced within the cell. The cell is a 3D shape, a sphere, a spherical shape. Therefore, the way in which they cut through could cut the mitochondrion at a different angle. And therefore, you need to understand that if you can clearly see the projections of the inner membrane, that is showing you the cristae, that is identifying the mitochondrion. So let's just look at the membranes then. So we need to remind ourselves that we have got biological membranes, that we're looking at the phospholipid bilayer, that there are proteins that are required. So in the glycolysis process, the last thing we ended up with was pyruvate, a 3-carbon molecule. Well, this is a larger molecule than one that can get through the phospholipid bilayer, so it requires a protein to help it get through. So the fact that it will diffuse through a protein within the membrane shows us that that is the facilitated diffusion. If it can get its way through from a high concentration to a low concentration through a channel, that would be a form of facilitated diffusion. So this is where your module 2 biological membrane content comes in. The next thing you have to note is the pyruvate is needed to get into that mitochondrial matrix. So the way it goes from the intermembrane space into the mitochondrial matrix is going against a concentration gradient. So therefore, active transport is required to get it from a low concentration to where there is a high concentration. So remember that when we talk about these movement of molecules, they will be expecting you to apply your knowledge from Module 2 and biological membranes. If they're telling you, for example, in my diagram, that there was a higher concentration inside compared to the intermembrane space, you know that that therefore has to be active transport. You would therefore know that ATP was required. So what happens when the pyruvate gets into the mitochondrial matrix then is what's known as the Links reaction. So the first thing that happens is pyruvate is decarboxylated. A carbon dioxide molecule is removed. That's going to take us from a 3-carbon molecule to a 2-carbon molecule. So we've got a 3-carbon molecule removing a carbon dioxide. We now have the 2-carbon molecule. We now have a coenzyme, NAD. We met that in glycolysis. Well, this is also going to be needed within the link reaction. So this coenzyme, NAD, is going to come along and accept hydrogen ions. And if it accepts the hydrogen ions and electrons, it is now becoming reduced NAD. Well, this is going to turn the 2-carbon molecule into an acetyl group. And then another coenzyme, coenzyme A, is going to come in and pick up the acetyl group, and it is going to transport it off to the next stage in the respiration process, the pathway that it needs to go to. So let's just remind ourselves, decarboxylation, the removal of the carbon dioxide, is going to convert the pyruvate to a 2-carbon acetyl group. So that's the first stage. Now what we need is we need oxidation to occur. So the pyruvate is going to be oxidated because the NAD coenzyme is going to come in and accept the hydrogens that have been removed, and that will form reduced NAD. So the coenzyme is reduced, therefore the molecule had to be oxidized. So that's how we end up with our acetyl group. And then what we have is our coenzyme A is going to come in and bind to that acetyl group. That is therefore going to become acetyl coenzyme A. And that acetyl coenzyme A is going to then transfer that acetyl group on to the Krebs cycle. So the key thing to remember about your coenzymes is they act like taxis. They can pick up specific passengers. NAD picks up hydrogens. Coenzyme A picks up the acetyl group. They will then take them and deliver them to where they need to go. So let's just remind ourselves, what was the input? The input was our pyruvate. 2 pyruvates go in because for every 1 glucose molecule, it is broken down into 2 pyruvate molecules. Then we end up with 2 acetyl coenzyme A's. We end up with 2 reduced NADs. And we have 2 carbon dioxides that are going to be released as a waste product. Now, if this were a autotroph organism, so therefore if we're talking about in plants, that carbon dioxide could be used in the Calvin cycle, the light independent reaction. In animals, this is not what we do, and therefore what happens is that carbon dioxide needs to be diffused out of the cell, picked up by the hemoglobin, and then transported within the bloodstream to the lungs for us to excrete it out of the body. Remember, the modules are always linked together. I hope you've liked this video. If you have, then please do click on the like button and subscribe to my channel. If you haven't already done so, then please do check www.aiqchat.com.